Functions of reagents used in acid-fast staining technique. ZnCF stain, also abbreviated as ziehl Nielsen carbol fuchsin stain. It is a primary stain. It is used to identify acid-fast organisms such as Mycobacterium tuberculosis and Mycobacterium leprae, which causes leprosy. Many acid-fast bacteria are not stained with the common stain like carbol fuchsin because they are prepared in aqueous solution. That is why they are not stained with the common stainers. These acid-fast bacteria require a stain that is prepared in phenolic stain, and ziehl Nielsen carbol fuchsin stain is prepared in phenolic solution, guys. As these acid-fast bacteria have a waxy coating on their surface, and phenolic stain solubilizes this waxy coating which is present on the surface of the acid-fast bacteria, and in this way cells get stained. The cell stained with ziehl Nielsen carbol fuchsin stain appears pink in color. The cell stained with ziehl Nielsen carbol fuchsin stain appear pink in color. Second one is the acid alcohol. It is the decolorizing agent. It is prepared from the combination of acid, that is 3% hydrochloric acid, and alcohol, that is 95% ethanol. Third one is 1% malachite green or 0.3% methylene blue. It acts as a counter stain. It stains the decolorized cell, and these cells appear blue or green in color. Mechanism of acid-fast staining. Acid-fast bacteria have a waxy covering on its surface, or we can say it has high lipid content in the cell wall. The cell wall of acid-fast bacteria is made up of lipids such as mycolic acid and glycolipids. Due to these have high lipid content in the cell wall, this cell wall has less permeability. So first it is necessary to increase the permeability of the cell wall, so the stain can easily penetrate in the cell. The permeability of the cell wall is increased by using phenolic solution and steaming. After the permeability of the cell increases, the cells get the stain. Now once the acid-fast bacteria gets stained, it does not decolorize even by the strong decolorizing agent and appear pink in color. But the non-acid-fast bacteria get decolorized easily and get stained by counter stain and appear green or blue in color. If we use malachite green stain, cells get the stain and appear green in color. And if we use methylene blue stain, cells get the stain and appear blue in color. Observation: acid-fast bacteria appear pink in color. Non-acid-fast bacteria appear green or blue in color. Now we will discuss about some important pointers regarding acid-fast staining. Permeability of the acid-fast cell is increased by phenolic stain, because phenolic stain have high affinity towards the waxy covering, and it solubilizes the waxy covering. For increasing the permeability, we use heat steaming, because steaming softens the waxy material and allow easy penetration of stain. Applications of acid-fast staining. Acid-fast staining is used in the diagnosis of Mycobacterium tuberculosis and leprosy.